Unmute. Okay, can everyone hear me? Perfect. Okay, so I'm very happy to introduce Professor Swanne Gordon, who is an evolutionary biologist and behavioral ecology professor at Washington University in St. Louis, whose research is built around answering the question of what generates and maintains diversity in nature. So to answer this question, she has worked with a number of interesting systems, including a classic system for studying local adaptation to environmental conditions, the Trinidadian guppy, and an emerging system for studying color polymorphism, the aposematic wood tiger moth. Specifically during her master's at McGill University, she compared introduced and local populations of Trinidadian guppies, and then went on to do her PhD at the University of California, Riverside, where she studied the rapid evolution of color and sex linkage in guppies introduced to novel environments. She then did a postdoctoral fellowship at the University of Jyväskylä in Finland, where she studied how selective forces maintain color polymorphism in the aposematic wood tiger moth. And on that note, Professor Gordon, please proceed with your talk.
Okay. Let's see. Try to set it up. See my busy desktop. All right, well, thank you so much, Aiden, for the introduction, and then also for our little chat that we just had. And it's nice to meet everyone, even if virtually. Okay, so I study diversity in nature, as Aiden nicely said, specifically how it evolves and is maintained across different levels of selection and also spatial scales, which is quite important because for the past few decades, a central goal of evolutionary biology, as we probably all know, has been to understand the origin and maintenance of variations or trait polymorphisms in nature. Now, in this respect, I always say a sort of paradox exists, because you can imagine that if any form or ecotype has an advantage over another, then it should technically go to fixation. But we don't technically see that everywhere, and there remain numerous examples of trait polymorphisms in nature across taxa, and none more so than when you think about color, which represents, in my opinion, one of the best examples of a functional and important polymorphism, probably owing to the fact that it's quite easily scored compared to other traits and hence tractable, but also because it's very widespread in nature across taxa, such as some of these examples that I list here. And so I study diversity and coloration in two main systems, as Aiden said. First is a rapidly evolving Trinidadian guppies, where in this system, we know that coloration and other traits rapidly changes when you remove predators from their environment, but we still know relatively little about the process. Now, because of time, I'm not going to say too much more about my guppy work, but I'd love you to stay tuned for some pretty amazing research lines we are doing currently and have just done related to the role of fluctuating selection, plasticity, and neuroethology in rapid evolution of sexually selected traits, and then the role of sexual behaviors on eco-evo dynamics. So I'm really excited about this stuff as well. However, today I wanted to tell you a little bit more about my second system, the aposematic wood tiger moth. And for the past 10 years or so, my colleagues and I, including the brilliant Johanna Mappes at the University of Jyväskylä and now University of Helsinki, has been asking the specific question: why do aposematic wood tiger moths come in so many different colors? Now, I like to call wood tiger moths, and actually I think many other Lepidoptera, a non-model model system of probably polymorphism. I think owing to the fact that they're not typically used to the extent that I think they should in this regard, but this is changing. So if you Google what a model system is online, you can get to a definition of a particular system or species of animal that has been developed over many years to powerfully answer particular questions. And now in this regard, there's certain criteria that you can use to justify it, such as is the organism easy to rear? Is it a convenient rearing size or operational size, or is it relatively inexpensive to operate? Now, when I started working with tiger moths, they were definitely not a model system. But over the few years, my colleagues and I have developed the knowledge enough that it's now seen as a good model system in the topics of counteractive selective pressures of predation, mate choice, immune function, and then recently, the topic of diversity in aposematism. So today I want to tell you about a few lines of research where we highlight wood tiger moth diversity, showing that mechanisms of diversity are rarely simple. Now, if you take nothing else away from my talk today, at least you could get that. The goal is to eventually synthesize all of these lines of research into a more integrative or generalizable or cohesive story. But before I go any further, I want to tell you a little bit more about aposematism. So as a term, it was coined by Poulton in 1890 and specifically means a combination of a warning signal, typically coloration, combined with some aspect of unprofitability, generally via a chemical defense. Now, aposematism is problematic both in terms of its origin as well as its maintenance. In terms of its origin, one can imagine that probably educating naive predators must have been pretty bad for the early educators, such as the schematic that I give you here with the monarch butterfly and the blue jay. So how did it become established in populations at very high frequencies? It's also problematic in terms of its maintenance because theory supposes that aposematism is expected to lead to monomorphism via positive frequency dependent selection. And this is because conspicuous coloration is going to confer protection to toxic prey only dependent on the high frequency of individuals which display the signal for efficient predator learning. However, despite this prediction, there are many examples of polymorphisms in aposematic systems, which in my idea or opinion provide us with an excellent opportunity to understand the selective forces behind diversity in these more challenging cases. So thinking about wood tiger moths, the system I work with, are they toxic? Well, let's check in a slow motion video that a couple colleagues of mine made in Finland. And so the wood tiger moth has two lines of chemical defense. They have an anal fluid, which we now know is targeted specifically towards insects, but then they also have a neck fluid, which is made up of pyrazines that bird predators really don't like. And when the bird tastes or squeezes the moth, it will release the neck fluid and the bird will shake its head in disgust. It will wipe its beak rigorously against something to clean it, which we know is a key sign of bird distaste. And it will drop the moth. We also know from a bunch of pilot studies that a moth can actually survive an attack of this sort. So it's actually a really beneficial defense. So wood tiger moths are aposematic or variable in coloration and toxic across their entire life stage, from egg all the way to adult individuals. As larvae, they're also toxic and they have a warning signal, which is a bright orange colored band over an otherwise black hairy body. The size of the band we have recognized via different research to mean greater efficiency against predators. And so the larger your orange signal, the better chance you have at not being attacked by predators. Adult females have continuous coloration, a continuous warning signal in their hindwings, ranging from yellow, orange to red coloration, whereas males have discrete color patterns. So they come in either white, yellow, red, or black hindwings. So for instance, in Finland, we have two hindwing morphs, the yellow morph and then the white morph. Now, typically, the wings are generally folded. And so the males will look quite similar, but when bothered or courting females, they open their wings and then they show their beautifully colored hind wings below. some populations across their global range have an admixture of both morphs, whereas others have just one. And you can actually see this represented nicely in their global distribution, where the male morphs are on the top panel and then the female morphs are on the bottom panel. And this is research that had been collected by the MAPIS lab over a variety of years. And so I'll point you to this area here where you see the bars in red. And so this region corresponds to the Caucasus Mountains in Georgia. And here we initially assumed to be the ancestral population of the wood tiger moths, which around the last glacial maxima, they became isolated as the wood tiger moths spread across the globe. Now, something that's really interesting to me, if this is the case, is that the system went from being seemingly sexually monomorphic. So both the males and the females have this red or orange-red coloration, Heinemann coloration, but then as they spread across the globe, then you see the existence and the coexistence of other morphs. So, of course, we expect selection on aposematism to be positive frequency dependent selection, with predators avoiding the most common warning signal in each locality, leading to fixation of one of the morphs in each of these spots. But yet you don't see that across the range. So the big question is, why in this particular system is there so much diversity when you expect positive frequency dependent selection? Now, one simple answer could be just that it hasn't reached complete fixation of the most common morph yet, right? So if that were the case, in some instances, oops, if I could get to here, in some instances, for instance, in Scotland, we've reached it, right? There's been complete fixation of the yellow moth, or in Estonia, almost complete fixation of the white morph. But then in other cases, it's just on its way to that. If this were the case, then we would expect to see evidence of positive frequency dependent selection in nature across the moth's global range. So we decided to test this. And how we did it is we performed an experiment where we exposed morphs to local predators in 4 different countries across their range. So Scotland, which is monomorphic yellow, males and females have the same coloration. Georgia, which is basically monomorphic red, males and females have the same color. In Estonia, around 95 to 98% of the males have only white coloration. The females are mainly red and yellow, and then there's a small percentage of males that come in yellow, so it's a polymorphic population. Whereas in Finland, white males and yellow males are combined with the yellow to red females, so another polymorphic population. We then monitored the abundance and community structure of the bird predators in each location in order to test whether selection by predators favors the locally common morph, which would be a signal of positive frequency dependent selection, or whether the community structure of the bird predators is associated with risk to a particular morph. By doing this experiment, which we just published this year, last year, we provided one of the best documented cases to date of a geographic mosaic of selection on warning signals at a broader spatial scale. And so we had 4 study locations ranging from monomorphic to polymorphic. We used these artificial plasticine wings with printed wings, and we used spectrophotometer readings to make sure that the printed wings matched really closely to what the real moth would look like. We had 60 predation transects with 20 white, yellow, and red moths pinned in a random block design every 15 meters. We left them for 6 days during the flight season. We recorded the predation events daily, and then we also recorded the bird community once at all sites. Okay, so here are our results, which is pretty busy, so I'm going to slowly run you through it. On the top panel, we have the local morph frequencies of the 4 populations. So again, Scotland, monomorphic yellow, Georgia, monomorphic red, and then Estonia and Finland, polymorphic populations for the 3 morphs across both sexes. If positive frequency dependent selection really was driving more frequency ratios in each locality, then we would expect that the associated predation risk should confer an advantage to the yellow morph in Scotland, the red morph in Georgia, and then depending on the local frequencies of the different three morphs, the advantage would be to either the yellow or the white in Estonia and Finland. So down in the bottom panel is our actual results. So this is predation risk here. Altogether, we observed a total of around 700 bird attacks on around 4,000 artificial moths. And we just, as I said, published this paper. And so in Scotland, we found exactly as we predicted. And so there is a survival advantage to the yellow colored hindwing individuals compared to the other two morphs, which matches the fact that the yellow are the locally common morph in that environment. In Georgia, we found the same thing, so an advantage to the locally common red morph. In Estonia and Finland, in Estonia, the white morph does have a trend towards having a slight survival advantage, but it's not significant. And in Finland, again, the pattern follows the proportion of moths. So another thing we also noticed is that predation pressure varied between and within countries across our transects, but overall predation pressure was higher in Scotland and then lower in Estonia, which gives us new things to kind of test and work out moving into the future. And so basically, we found positive frequency dependent selection, which was fantastic, and we were really excited. However, in looking a little bit more deeply into the results, it alerted us to another interesting issue. So when we principal component grouped all of the bird predators by their feeding tactic in order to use it to kind of describe the predator community, which we could then link to morph survival, we found something really cool. So this is estimated attack risk. And basically, for instance, looking in Finland, so we found that the yellow morph is favored or attacked less in communities characterized by paridae or great tits and blue tits, whereas in contrast, an opposite result was found in Scotland. So in the same predator communities, so tits, great tits or blue tits, the yellow morph is actually attacked more in Scotland. And so this is really interesting because it tells us that differences in predation pressure at the population level might actually be quite important. It alerted us to another interesting issue. Now in 2014, there was a paper published by Ossi Nokalainen and colleagues that found that yellow males were significantly less attacked than white males in southern Finland, which is where we had our transects. But in our study, it was actually the yellow morph tended to have more attacks, not less. And so we were confused by this. Why weren't the results repeatable across years? But then we remembered something that's kind of cool. So interestingly, the frequencies of yellow and white morphs varies in southern Finland in a biannual cycle. And the yellow morph was more common during the year that Ossi Nokalainen and colleagues did their study, whereas the white morph was more common in the year that we did our study, which provided even more evidence bolstering the fact of the role that frequency-dependent selection might be playing in attack risk to these morphs. And it also tells us that temporal fluctuations in local predator-prey interactions could also plausibly be important. So I decided to delve a little bit further and test positive frequency-dependent selection in this system, but on a finer scale, using a more stable population, so one where the morph ratio will not shift, and then using an actual real moth so that I could include the chemical defense to tease apart better that effect of a local predator. Most studies in frequency-dependent selection in aposematic systems, or even outside of aposematic systems, focus broadly if you really look in the literature. And that means that they may actually miss information on the actual predators, their behaviors, and variations in their responses that may provide more mechanistic insight as to the role that frequency dependence might play in warning systems. And so I performed an experiment with my colleagues, Professor Emily Birdfield-Seal and Professor Johanna Mappes, where we used an indoor exclosure, where we could see the bird, but the bird couldn't see us. And then we used recently killed and frozen thawed moths so that we could have fresh defense or chemical defense included in this while controlling for the behavior. We had 4 treatments where we had a balanced control, so equal numbers of whites, yellow, and red moths. And then we had a white bias treatment that had 12 white moths with 6 yellow and 6 red moths. And we also had a yellow bias as well as a red bias treatment. We tracked the predator, the local predator, which we used great tits, behavior, so whether they were cleaning their beak after eating, after attacking a moth, or their drop behavior, whether the taste was really horrible. Also, the bird size, its age, which is a proxy for its experience, right? Whether it's juvenile or an adult, and then the order of attacks. We ran the experiment until the bird ate half of the moths in the treatment, or until 2 hours have passed. And we've just recently published this paper in American Naturalist. And so then we wanted to see, is there positive frequency dependent survival selection? So basically, who gets eaten? And we actually found some really cool results. So I'm going to run through it pretty quickly. When we look at the balanced control or equal numbers of morphs ratio, we find no difference, no significant differences in attack risks. We also found no significant differences in the female bias treatment, where the red morph was the more was the locally common morph. In the white bias treatment, we did find positive frequency dependent selection. So the more common morph, the yellow moth, had a survival advantage. We found a slight one for yellow, but it wasn't significantly different from the males for the yellow bias treatment. And then we were puzzled about the females. What's happening there? Does frequency dependent selection not extend across the sexes or to the red individuals? But then when we analyzed all of the data in one, we find that yes, the reds as well have positive frequency dependent selection, but only because there was an apparent benefit to being common in comparison to their much lower survival when rare. And so you can see that when the females are rare, when there's only 6 of them in the white bias or the yellow bias treatment, for some reasons, the birds would eat them. They would really search for them. It wasn't a mistake. They were going for those females, compared to when there were 8 individuals, so more colored females in the arena that the bird was able to look at, or when there was only 6 of them in the 2 other trials. So this was really, really interesting that we were able to find it at this low scale. And I won't talk about it today, but we found that not only do we have positive frequency dependent selection present, but it was very much complicated by predator age, predator sex, and also variable responses to prey profitability based on their levels of defenses, meaning that however important positive frequency dependent selection might be in aposematic systems, other selective outcomes might be common and likely dynamic. And they underline the need to kind of understand predator foraging in order to better examine how behavioral interactions might shape evolutionary outcomes for these sorts of systems. So, positive frequency dependent selection, which we find on both a global as well as a more fine scale, should lead to fixation. So, what does that mean for our global polymorphism? It doesn't technically explain the patchiness that we see in our global range, where we have instances that might stretch across a predator's range, but where you have vast differences in the ratios between the males and the females in the wood tiger moths. So it's puzzling. So how is polymorphism maintained in the face of positive frequency dependent survival selection? The short answer is we don't fully know yet, and we're working on it. However, a past experiment may hold some clue. And that past experiment was inspired by the research that I did in guppies during my master's and my PhD, where a big controller or maintainer of polymorphism in the guppy system is an interplay between natural and sexual selection. And so I decided to do an experiment where I thought maybe mating selection, maybe negative frequency dependent mating selection could provide the answer that will maintain polymorphism in this system. And so we performed an experiment in an outdoor enclosure that was around 3 meters high and 20 by 30 meters, where we tested whether negative frequency dependent selection in the context of sexual selection could balance morph ratios and allow rare morphs to still flourish. And so we did it under 3 different morph frequencies in central Finland. This time, we included only the yellow and white male morphs for Finland as the ratio. So we had a white bias treatment, a yellow bias treatment, and then a balanced control. The red females were used as the females, which we tethered, if I could get my pointer, we tethered to these large styrofoam boxes in each of the cages so that they could still fly and move around, but we were able to keep them in place so that we could see which males were approaching the females and which males would eventually mate with them. And then afterwards, we could easily recapture the females so that we could bring them to the lab and raise the resulting offspring. And you can check out these corresponding papers if you wanted to learn a little bit more about this paper. But basically, we expected negative frequency dependent reproductive selection. And unfortunately, we found positive frequency dependent mating selection, which was extremely puzzling. So looking at individual relative fitness, which is the number of offspring for each male compared to all the other males in that particular enclosure, for the white bias treatment, the balanced treatment, and then the yellow bias treatment. And you can see that in the white bias treatment, the locally common morph, the white morph, had the advantage. And so they had the higher reproductive success of resulting offspring. Same for the yellows and the yellow bias treatment. And there was no significant difference in the balanced ratio treatment. So we were completely confused. Now we don't even have one instance of positive frequency dependent selection that's supposed to drive something to fixation, but we also have it on the mating end as well, positive frequency dependent reproductive selection. And so we were puzzled even more as to explaining the patchiness in our system until I came across a paper that had a word argument or thought argument that potentially, if considered under a spatial context, where in each local environment across the organism's range provided an advantage to one ecomorph or type over another in each environment, combined with a little bit of gene flow, could be enough to maintain polymorphism. So we next integrated our data from the mating experiment with known estimates of predation against both morphs in a simple spatial model in collaboration with the brilliant Hannah Coco. Now, this was amazing because this trade-off was particularly unexplored in a case where both natural and sexual selection are potentially positive frequency dependent selected. And never before had variation in frequency dependence across fitness components been considered in this context. So it was really, really cool paper. So basically what we did is we had 250 white and yellow moths randomly spread across patches, which gave us our initial white and our initial yellow values. We had 60 habitat patches, which corresponded to the varying predator communities and the attack risk to either the white or the yellow morph from the data that we had collected in the field. We then tracked morph frequencies over time as it went through this predation risk. Afterwards, we made each patch equally productive, so around 50 offspring leading to the next generation. And who left the most offspring was based on our findings from our mating experiment, where if you are the locally common morph after the predation event, then you would lead more offspring to the next generation. We ran the model for around 1,000 generations and then included different levels of dispersal or gene flow in order to see if we could find polymorphism or if we could maintain polymorphism. And we were able to do so. So looking at the proportion of yellow morph here, at 0, it means the white morph has gone to fixation. And at 1, it means the yellow morph has gone to fixation. And you can see that we can maintain polymorphism along this line as long as dispersal was limited to just a little amount. And so that was just an absolutely fantastic finding. Don't mind about the different symbols. It's just that we had multiple mating in our mating experiment, and we also wanted to put in if one morph left more offspring than another when multiply mating in multiple paternity, then would that result change? And it really didn't. Okay, so the next thing we wanted to have, let me just check the time. The next thing we wanted to ask is that, yes, now we can maintain polymorphism, right? But only if technically these moths do disperse. So do they move? Is there gene flow from patch to patch or even within a local patch in the system? And so I decided to do a field Mark-Recapture, an intensive field Mark-Recapture on these individuals. Now, when I first proposed this to my group at the time, they were like, nope, you can't do it. We could maybe catch them like we do all the time, and we can census them by setting out pheromone traps. But doing a Mark-Recapture on a flying insect is going to be almost impossible. But of course, I come from the guppy world. So I was like, yes, challenge accepted. We love, like taking them out of the stream and putting them back in the stream and moving them around. And so I was like, yes, I can do this. And so I wanted to ask the simple question, well, not so simple, but I wanted to ask the question, is the population structure of wood tiger moths in central Finland influenced by sex, survival, or time-biased movement? And I did this with a variety of co-authors. But because it was really tough to do a Mark-Recapture with flying insects in the field, I had a lot of help from a variety of individuals, colleagues, lab members. It really followed the whole thing, it takes a village, but now in this case, it's like it takes a village to do science. And because a picture says 1,000 words, these are a couple of pictures of my time in the field during the Mark-Recapture. And so it's my ode to Finnish summer. You can see that this is July and August in Finland, and we are wearing long sleeves to all the wonderful mosquitoes, and they were pretty bad. Great colleagues, and also being super mamas in STEM. And so in this picture, I'm actually pregnant. And then 2 years later, my son could actually come out to the field and help with collection. So it says a little plus about having children when you're academic. But I really want to thank all of these people. And so the questions we wanted to ask is, do moths disperse, and is it biased? So from lab and field surveys, we assume that the population probably has male bias dispersal, because when the females eclose, they are filled with all of the eggs that they will eventually lay, so around 200 to 400 eggs. And you can see that it's extremely cumbersome for a female to fly. But once she starts laying a couple of the eggs, then her flight becomes a lot better. And so we anticipate there to be male bias movement. We anticipate that the males potentially move more and that they do move first, and that the females may move more after mating and that they start dropping some of their eggs before they start searching other places to lay the rest of the eggs. We also find from our censuses that we have collected that we knew they would be at low densities. So we knew that we really needed to do an intensive Mark-Recapture to be able to get enough recapture probabilities or time points that we could look at the recapture profiles. So we did this recapture over 2 seasons in central Finland. We had approximately a month each season where we had 800 transects, where we measured around 6 to 12 hours per day plus ground search. We collected all of the data for personal man hours, weather, habitat variables. We did a bird predation survey each time that we did the season. We had individual capture and recapture information. We also took pictures of the individuals that we captured in the field so that we could have morphological structure profiles. And then we took a small leg clip from every individual where then we could look at relatedness over time. And so just some descriptive information. You don't need to look too much at the data itself. But we did find that our hypotheses from what we had done in our censuses and also at our lab studies over the years was right. So the males do move more. The white males a little bit more, while the yellow males seem to stick closer to the females. So showing this difference in their behavioral strategies that match to a flying experiment that we had done a couple years and that we published recently. We also find that the yellows and the females cluster around their habitat vegetation. So they cluster around the transects, whereas the whites are much better at spreading around that path and searching for females. The whites do fly first, the females emerge last, and that the peak season is really driven by white males, which lead to the stable bias in white males over time. Looking at recapture differences in movement before the morph, between the morphs, again, it wasn't significant, but we had a slight trend where the white males move more, but we do have a difference in that the females significantly move less. We did find a significant interaction between morph and wing area, such that the bigger your wing as a yellow individual, the less you move. And this might be related to predation in some way, but we're still needing to tease this apart in an actual experiment. And then we wanted to ask, are there seeming differences in movement that are influenced by genetic structure? And so we did a total of 182 individuals from 2 years that we genotyped at 20 microsat loci. And then we found this result. So I just got this graph, so it's not very pretty, but basically what it says is kind of what we had anticipated or what we had hoped for, but even more than we thought. And so the majority of individuals from year to year are unrelated, which lets us know that that recapture data that we're showing that they're moving a whole lot is also shown in their genetic profile. We do also see that the females are more related. And that also tells us that the females are moving less. So they're staying in a patch, laying their eggs, and the next year you can see that the females are still there, whereas the males are moving themselves out as soon as they eclose. The years are similar, so it's not a fluctuating population, which we knew because we knew that graph that I showed you of central Finland is very stable, unlike southern Finland that has its biannual cycle. So basically, male and morph bias movement exists in wood tiger moth Finnish populations. And now we're doing some more spatially as well as genetically explicit modeling to examine which of the traits we collected, morphology, weather, habitat predation, etc., best influences survival and dispersal. So my broad conclusions is that positive frequency dependent selection in both mating and survival combined with dispersal can lead to polymorphism in the wood tiger moth. So basically, multiple factors contribute to diversity, not just frequency dependent selection, but a variety of others that I've shown you piece by piece in these different lines of research. And I think that should be the key in any of our research where we're kind of looking at these things, to not run from the complexity, but to embrace it and step back and see how we can actually do this at a broader scale. So where has my group or my lab gone from this research? Well, a variety of different directions. We know that the theoretical model that I did with Hannah Coco to show the maintenance of polymorphism doesn't match perfectly well, again, that patchiness that we see across the scale, the global range. And that could be that because we were missing a variety of key parameters. And one of them could be the relationship between life stages. I think we're all guilty of this across our systems to some respect, where we focus heavily on either an adult stage or we focus heavily on a juvenile stage, and we'll publish separate papers. But not often do we think that the connection might actually be strong from larvae to juvenile and back and forth, to adult and back and forth. And something that kind of brought us to this conclusion was a master's student of mine, Diana Abondana Almeida, who's now doing her PhD in Germany, did an experiment to examine the role of plasticity in the variable anti-predator larval signal of the babies here. So how large or how small your signal is. We knew that warning signal in larvae as well as adults is very strongly genetically controlled. We wanted to see if there was an environmental component. And so she stimulated a predator attack from the larvae were really tiny or really young, the first few molts, all the way up to adulthood. And then she looked to see if there were any changes in the reaction norm of siblings that were reared under the predation environment or ones that were just kept normally or reared normally. And we did find that. So we found that there was plastic as well as genetic control of the warning signal of larvae. Why is this important? Well, a colleague of mine, Professor Carita Lindstedt-Carricksella, and I found that there is a negative phenotypic as well as genotypic correlation in warning coloration between larval signals and adult signals, which is really cool. And so if you are a large signal larvae, you have a higher probability of becoming a less efficient against predators female wood tiger moth morph. So you have a high probability of becoming a yellow-orange morph. Whereas if you have a low efficacy warning signal against predators as a larvae, you have a higher probability of becoming an adult female that will be better efficient against predators. Same thing on the male side. So if you have a large larval signal, you have a greater probability of becoming a white male. Whereas if you have a small larval signal, a less bright signal, a less large signal, you have a higher probability of becoming a yellow individual. So what happens or selective pressures at the larval stage can potentially inform the ratios of morph or polymorphisms at the adult stage and vice versa. So a PhD student of mine will now explore the role of plasticity across the life stages in maintaining diversity of sexually selected traits for their lab and in the field for their thesis. But one thing that we came up upon was that we needed a method to individually track organisms in the lab as well as the field. Of course, if you pull a leg like some individuals do, once they molt, then, it's back, right? But also if you put a little paint mark like we do on the adults, again, when they molt, it's gone. And so I developed a new method of marking individuals across life stages that was inspired by my work with the Trinidadian guppies. So to mark guppies, we use elastomer paint and we inject subcutaneously just a small blob of paint underneath the skin, and that travels with the individual throughout their lifespan and they rarely lose their marks. And so I did the same thing in wood tiger moths. I injected very small larvae with a small blob of elastomer paint in an area where I knew where it would pop out as an adult. And then I tracked that paint throughout the life cycle to check and make sure that it would remain. And so here's just, so the paint, some of it fluoresces. And so this is under the blue light of an individual where you can see the pink spot in the adult, and you can see the pink spot in the larvae, even shining through the black and hairy body. Sometimes even without the light, you could see the pink spot in these individuals. So now we can track individuals both in groups as well as in the field, which will be really useful for doing this study. Another thing that the model was also missing was information regarding the inheritance or genetic basis of coloration. A pedigree that we had over 10 years allowed us to identify that male hindwing coloration actually had a simple Mendelian inheritance. And so the lab has now bred selection lines for white homozygote, heterozygote, as well as yellow homozygote individuals in the MAPES group, which we as well can also use to now link phenotype to genotype in the lab and the field. And this was also inspired by work done by another master's student of mine, Sini Berdulat, who ended up doing an experiment to show hybrid advantage in wood tiger moths. So basically individuals that come from a genetic lineage that combines both white and yellow in Finland have a reproductive advantage over individuals that come from purer lines. So this is actually really, really awesome work. Okay, I'm also going to be doing an urban ecology project in butterflies and moths, which take into consideration socioeconomic factors. And so this is some really cool work that I'm excited about, as well as some really awesome guppy ideas and experiments related to diversity in nature. Okay, so that's it. And this may have seemed like a whirlwind of studies trying to explain just one important and potentially simple phenomena. Why does diversity exist, and why is it maintained in nature? But hopefully it highlights that, the wood tiger moth system and other Lepidoptera invertebrates might actually be pretty ideal systems for studying theory related to polymorphism and variation, especially since we've been able to do so much in the system in so little time. And that be, again, the take-home message that diversity is ever rarely simple, and we should really embrace complexity in how we try to understand it. With that, I want to thank my funding sources and people that have provided collaboration and advice to me over time. I also want to thank my old lab members or old collaborators or research interns, in addition to my new ones. And so come join us. We're a fantastic team. And, we really preach open inclusiveness and equity in our group. And we're always looking for more people that also agree with that. And then finally, I've started doing this ever since last summer, with thanking the many named and nameless heroes that have paved the way for me in education. I usually give a big speech here, going into things and showing data and statistics and things like that, or at least mentioning it. But I feel that there's nothing more left to be said about systemic racism or systemic bias in academia that hasn't already been said, that hasn't already been published, that hasn't already been discussed, right? And so the statistics for our field of ecology and evolution is quite stark. So there's only 0.3%, for example, of Black faculty in our field, and massive underrepresentation across metrics in funding, in publications, in participations in collaborations. And so we highlight this and more in a co-authored paper that we just published in American Naturalist, led by the wonderful Meg Duffy, and a paper that we actually just put out in review in Ecological Monographs, led by the fantastic Dr. Nahima Harris. And so I urge you to go and look out for these papers. And I think this fact or these disparities are superbly unfortunate and a huge loss to scientific progress, innovation, and also advancement. Because like I like to say, only when the broad diversity of humanity is fairly represented can science truly appeal to our society as a universal knowledge. And with that, I'm going to leave you with two quotes from two of my favorite people, one taking too soon in his time, a Canadian researcher, and another one, my favorite author. And with that, I'll take any questions and comments. Thank you.
People can raise their hands or unmute themselves or something. Somebody has a question here. I could retry this. Can you all hear this? Can you hear this?
Sort of.
Okay, I'll try to speak clearly. Very interesting talk, and very fascinating. Your comparisons are all about relativeness, and I want to ask a question that goes beyond that. Is it possible that the low frequency morphs may be maintained because predators, once it's been established that this frequency, high frequency morph, avoid that species of butterfly altogether and choose alternative prey, so that even the ones that aren't the right color still get benefit in their fitness because the predator is switching to an alternative prey. Is that a possibility, or is anybody that kind of question?
Yeah, so I'm going to try and answer. I'm not sure that I heard it really well, but I heard, I think, enough words that maybe I can piece together the context. So this idea of the alternative prey is actually quite interesting. And the reason why we didn't include alternative prey, for instance, in our fine scale experiment is because wood tiger moths are toxic. And so a bird will eat them. They will readily eat them, especially if the females, they have a way of maneuvering the female who's very toxic, right? So that they could kind of pick her head off and then eat the eggs in there that's high nutrients. But if they are provided with an alternative prey, something that they don't have to deal with the pyrazines and other things, they will eat that prey. And so wood tiger moths are palatable, but they're toxic enough that a bird might choose a more palatable prey than them. How that would work in terms of our results. And so we have done experiments in the field with live as well as the plasticine moths, showing that the birds will attack both alternative prey that are around, as what we've watched of their predation event anyways, as well as the pinned moths. And so we think that what we're showing is actually an artifact of, frequency dependent selection, especially because in the field, the birds do have the ability to choose something else, whereas in our fine scale treatments, they don't. And the fact that we find the same results across each of these spaces, including another experiment that I did in an outdoor enclosure that had other insects and things for the birds to eat as well. So the fact that they all show similar results seem to point or give more credence or evidence to positive frequency dependent selection being important. But I think what the bird community data shows, as well as the specific predators themselves and how the strategies with which they attack the wood tiger moths show that there are other factors in play as well. So you're exactly correct. Something that we did not show or did not show today and that we have not published yet is that we took measurements of all of the potential alternative butterfly and moth communities in each of these sites that we did that experiment, because we wanted to say something about potential generalization if there were more palatable or unpalatable insects for the birds to eat in each environment, which may have given more or information or reasons behind the finding of the varied predation pressure, as well as our actual results that we're still teasing through. And we've also done a couple of experiments looking at generalization across these moths, as well as learning that might start to tease apart some of those different factors. So I hope I've sort of answered your question because I couldn't hear it very well, but if not, then please, you can say something else.
Let me just one follow-up, because I think. So, do all morphs benefit as long as at least one of them causes predators to avoid those ones?
Sorry?
Do all morphs benefit as long as at least one of them causes predators to avoid the moths?
Yeah, so so that was the work that we did with the generalization, right? So if you have eaten or had some experience with one morph, does it change the way that you would attack or eat another morph at a different time? And we find that the moths are the predators, at least great and blue tits, are able to keep them quite separate. I did do an experiment in the field where I did a bird predation experiment where females were included in it or they were not included. And that seemed to change the bird's attack order over time between the white and yellow male morph. Now, the problem with that study is because the females have these juicy eggs, right? And the males do not. And so it's hard to kind of tease apart what that really means. But I think there might be something there that even though we haven't been able to find that they do generalize across color or pattern, that they might actually depend on their experience in one area, you know, with a locally common morph, might be able to shift their behavior or strategy in another area. And so these are all questions that I think we're looking out for, we're hoping to be able to answer moving forward.
I think given the time, we should thank Dr. Gordon again, but if people want to hang out and talk some more, then they can also do that on the same Zoom call. Okay.
Thank you.